Hello my dear students and welcome back to Excellence Batch, and I am your Diksha ma'am. In the previous class we have discussed about fertilization. What are the events near the fertilization, what are the changes that occur in sperm, and how does a fertilization takes place. So today it's time to move further, but let's check it. How much have you studied? So for that I have brought some questions for you guys. Let's solve them. First, what is the site of fertilization? Second, receptors of sperm binding are present on which layer of oocyte, right? Capacitation occurs in, that means at which place capacitation occurs. Rather than in, I think I should write at. We have to tell the place where capacitation occurs. Enzyme that digests zona pellucida, and which centriole of sperm enters the ovum. Very simple question. They are kind of a quick revision for you. First is site of fertilization is as simple as it is the ampulla of fallopian tube. Ampulla of fallopian tube. Next, receptors of sperm binding are present on. Where are these receptors present? Definitely on the layer known as zona pellucida. So on zona pellucida we have ZP receptors which bind with the sperm, and then the recognition takes place, and hence we say fertilization is species specific. Next, capacitation occurs at female reproductive tract, not in the male. Why? If it occurs in the male, the sperm will not be viable enough, because after capacitation it has a life of around 48 hours, and if it would have occurred in the male part only, then the sperm will never survive after that. The enzyme that digests zona pellucida is zona lysin, also known as acrosin. And the last, centriole of sperm that enters the ovum is always proximal centriole. So proximal centriole is a centriole that enters inside ovum. So let's score yourself out of 5. So what is your score? You have to tell me in the comments, right? Okay, so that's about a quick revision, but let's get started with the further plan. But before that, let me explain you fertilization one more time. Let's have a recap so that next things should be easier for you. So what was happening? The oocyte has been released. Oocyte has been released from the from the ovary. It has entered in the fallopian tube. It has number of layers like zona pellucida. It had corona radiata. Now the sperm has entered inside the oocyte, and sperm has entered digesting all these layers, and it has entered. Now the sperm has given, as soon as the sperm enters the cytoplasm, this oocyte gets converted into ovum. This is oocyte. As soon as the sperm touches the cytoplasm of oocyte, it will complete its meiosis 1, meiosis 2. Meiosis 2 is completed and it has become ovum. Now it has another nuclei which is of the sperm. Both they are going to join, and hence they are going to become a diploid cell-like structure known as a zygote. So now this zygote has formed after fertilization in the ampulla of your fallopian tube, right? Now let's see what will happen next, because from zygote the entire human being is going to form. From zygote entire human being is going to form. So definitely we need to increase the number of cells. I am multicellular, right? So when a baby is formed, it's a complete baby with organs. So how the baby with that organs are formed from this just single cell known as zygote? That's the beauty of nature. So let's see what happened. So this zygote, it will undergo some division. It undergoes some mitotic division, and these mitotic divisions are a little different from mitosis. They are special division known as cleavage. They are like a mitosis, but they are not exactly mitosis. Like, so they undergoes mitotic like division, right? Or mitotic like division will be better to say, because they are not exactly mitotic division. That is not a mitosis. It's like mitosis. Okay, so the name of this division is known as cleavage. So zygote will undergo these divisions known as cleavage so that it can divide further and increase in number of cells, right? Because just single cell, how it is going to be a human? Definitely we need a number of cells, right? So now next stage that occur after fertilization is cleavage, and as I have told you, it is like mitosis. You will never find cleavage divisions anywhere else in your body. The cleavage only occurs in the zygote and runs up to only early embryonic stages, right? So it occurs in zygote. It occurs in zygote until early embryo. Until early embryo. In fact, not in the entire gestation period the cleavage is going on, right? The cleavage does not occurs for the entire gestation period of 9 months. It only occurs in the starting of development right after fertilization, right? Now, what is so special about them? How it is different from mitosis? Like mitosis, the cell number will increase, but it lack the growth phase. It does not have growth phase. If it does not have growth phase, remember there is like a growth phase, interphase, right? Botany, cell division. So it does not have this phase. If it does not have this phase, definitely the time of division will be less. The time that one cycle, mitotic cycle was taking was much. Why? Because growth phase take a lot of time, but here growth phase does not occur. So that's why these divisions are very fast as comparison to the mitosis. So they are very rapid division. Another thing, the cells that are formed after mitosis, you call them as daughter cells, and daughter cells are of the same size of the mother cell. This is what you do in mitosis, right? So what we see in mitosis, there is a cell, mother cell, it will divide and the cells will be of the same size. You can see the size of the cells are same. But what happened in the cleavage? In cleavage, if this cell is a mother cell, which is a zygote, if this is a zygote, the cell will divide, and the daughter cells will be smaller than the zygote, right? How? For example, it has divided with cleavage and the size will be the same, and you can see these two cells is a daughter cell like these. These cells will be of the smaller size as comparison to the mother cell. So here mother cell is zygote and daughter cells, they are known as blastomeres. What do you call them as? Blastomeres. So the mother cell here is zygote and daughter cell here are known as blastomere. Here these are known as daughter cell in mitosis, right? Here they are blastomere. So here we got to know one thing, that the blastomeres are smaller in size than zygote. Now why is it so? Because growth phase is like it equivalent to like it does not appear, right? Growth phase is absent here. So when the cells are dividing, the cells are only making sure that their nucleus will divide, this cytoplasm should not divide. So here the nucleus is rapidly dividing but cytoplasm is not dividing, the cytoplasm is same. For example, if I say whatever quantity of cytoplasm is present in zygote, it is present same in both the cells. So cytoplasm is not growing, the nuclear divisions are occurring, the nuclear material is growing, right? So here nuclear material is increasing, it is dividing, right? But cytoplasmic or cytoplasm is not increasing, it is still the same. So if you will see nuclear to cytoplasmic ratio, it will be higher in these cells. it will be higher, because cytoplasm is less, nucleus is more, so ratio will be higher. If you do the opposite, ratio will be low, right? So these are the features of your cleavage. So in cleavage, the blastomere size remains smaller and smaller, right? The size of blastomere is always less. The blastomeres are always smaller than the zygote, right? So now what will happen? Now for example, this is your fallopian tube. This is the uterus, okay, and this is the ovary. This is the ovary. Now as the ovary has done ovulation, it has released the oocyte. Now oocyte in the ampulla has met sperm, the fertilization has taken place. Now after fertilization, the zygote has been formed. So what is this? Zygote. So we are assuming the zygote is formed. So after 24 hours of fertilization, the cleavage starts, right? So what is cleavage? These rapid division, mitotic division, where the cell number will increase, but it does not show growth phase, so it does not have a lot of cytoplasm. Whenever the cells will divide, they will have less cytoplasm but more nucleus. As a result, they don't have cytoplasm. As comparison to daughter cells of mitosis, the blastomeres, they will be smaller in size. Right? Now, so this zygote has been formed after fertilization, but after fertilization, after 24 hours of fertilization, first cleavage will start, and first cleavage is meridional. Meridional means like this, straight, vertical, right? So first division will occur after 24 hours of fertilization, and it will cleave the cell like this, and two cell stage will be formed. So here you can see this is 2 cell stage. After 2 cell stage, 3 cell stage will be formed, and second division will be equatorial. You can see here this cell remains the same, this one is dividing, so horizontal, like transverse division will occur. Meridional means straight, vertical division. Like I have 2 cells, if it is cleaving the cell like this during division, it's vertical. If it is cleaving like this, it is equatorial. Now 3 cell stage has been formed. I am writing 3 cell here, right? After 3 cell stage, immediately 4 cell stage will be formed like this, right? 4 cell stage will be formed. After 4 cell stage, 8 to 16 cell stage will be formed. 8 to 16 cell stage will be formed, and this stage has a name. It is the early embryo known as morula. So morula is generally formed at 3rd day after fertilization. So after 24 hours of fertilization, that means on 2nd day your 2 cell stage has been formed, and after 24 hours of that, the morula will be formed on the 3rd day. Like imagine today the fertilization has took place, this is 1st day, 2nd, and then our day after that, 3rd day, morula will be formed, and morula is still present in fallopian tube. You can see this is still present in fallopian tube, right? Now this is your uterus. Let me draw it. This is the uterus, okay, this is uterus. All these stages they are taking place in fallopian tube only. Now this morula, it will leave this particular, what you say, fallopian tube, right? But before that, let's see how morula looks like in detail. Morula. So morula is early embryo. It is 8 to 16 cell stage and looks like a mulberry, like a shahtoot, right? It is mulberry shaped. So you will see the size, if you have seen carefully, whatever the size of zygote was. So here zygote is looking like a little small structure. Let me just make it a bigger. The size of a zygote and size of a morula is same. Why? Because the divisions that are taking place are cleavage. So the cells of the cleavage, known as blastomere, they do not have cytoplasm. They are smaller in size. As a result, due to cleavage, the size of morula and zygote is same. The size of morula and the zygote is same. Why? Because cleavage are happening and blastomere size is not increasing. So, it remains the same, right? Just like we have one ball. Okay. For example, I don't have ball here. I'm so sorry. We have one ball and imagine inside that ball, small balls are growing. Right. So, the size of the ball is not increasing, just inside of that hollow ball, you know, the number of cells, they are increasing. That's it. This is how it is. Fine. The ball size is still same, like you have a Costco ball or a leather ball. Inside of that, number of cells are present. So, earlier this ball was also zygote, but inside that the divisions are happening, taking place. So, the number of cells increase, but the size of the entire ball is still same. Like if I say the size of zygote is 0.1 mm, the size of the morula is also 0.1 mm in diameter. Okay. So, here the size of zygote and morula is same, right? Now, how does it looks like? So, if you'll see on the outer periphery, you will get these cells, and these cells, they are very tightly packed because they have tight junctions in them. The junction we have done in the epithelial tissues, so they are present here. These cells are very tightly packed. The entire ball of morula is highly compactly packed, or I say the cells in the morula, they are highly compactly packed, right? Why? Because zygote still had zona pellucida. Zygote still have intact zona pellucida. So, this zona pellucida persists up to morula stage. So, see, that's why this is another reason the size is same. That's why when a number of cell will increase, it will create a pressure. So, we say all the cells are very tightly packed because it also have a layer of zona pellucida around it. It has not gone anywhere, right? So, it has zona pellucida around it and the cells are very tightly packed. So, this is known as stage of compaction. The morula is known as stage of compaction. Outer layer of cells, they have tight junction in them. The inner cells, they have gap junctions in them. The inner cell, let me draw them. The inner cells, they have gap junctions in them. So, like imagine these are gap junctions. Okay. So, they have gap junctions between them. Alright guys, so it's a stage of compaction because it's still covered with zona pellucida, number of cells have increased and they are very compactly, tightly packed. Compact means tightly packed, that's why it's known as a stage of compaction. So, still the zona pellucida still survives up to this stage. Okay. Now, what will happen next? Now, morula will try to come inside uterine cavity. It will try to come inside uterine cavity. Now, it will do mitosis. It has left cleavage because if it will keep on doing cleavage, the size will never increase. And how will you make that 3 kg baby? Right? If you will keep on continuing cleavage division, the size of cell will never increase. The size will equivalent to zygote only for throughout life. But we don't want to do that. We want to make a big large size baby. So, what we do here, or what nature does, it starts mitosis. And when it starts mitosis, the size increases and in fact the number of cells increases. So, here the stage which you see is a 32 cell stage embryo. Now, this is 32 cell stage. After mitosis, 32 cell stage has formed. Now, because mitosis has occurred, the cell size increases. Now, zona pellucida cannot persist. Now, zona pellucida start to break. Zona pellucida, it starts to break. Now, the cell or this embryo will start to move outside. It starts to move outside. So, it will break the zona pellucida. It will break the zona pellucida and the embryo, it starts to move outside. So, here zona pellucida breaks. Zona pellucida break. I am writing ZP. ZP breaks. It is all happening in uterine cavity. An embryo escape. In the meantime, there is one more thing that's happening. Uterine cavity is glandular. It is secreting something. So all the nutrient secretion, they are entering inside the embryo. All the nutrients, they are entering inside the embryo. As a result, a cavity will be formed inside the embryo. A cavity will be formed inside the embryo, and embryo will take a new shape. It will become a new embryo. The 32 cell stage, when it is escaping outside, at the same time the nutrients are also entering from the uterus cavity, from the glands of the uterus, and as a result a cavity will be formed inside. So this will look something like this, right? So some cells will form a wall, some cell will gather here like this, right, and this cavity will be formed, and the name of the cavity is blastocele. Blastocele. And this embryo, 32 cell stage embryo is known as blastocyst. This embryo is known as blastocyst. So blastocyst is 32 cell stage embryo with a cavity. And what is the name of the cavity? Blastocele. You cannot call this as blastocyst. Why? Because it does not have a cavity. The cavity is present in this stage. Now the outermost cells, they are known as trophoblast. This stage has been formed on around 4th to 5th day. So when it is formed around 4th to 5th day preceding fertilization. Okay, and these inner cells, because they are present inside, the name of these cells is inner cell mass. Inner cell mass. Inner cell mass are the cells that will form the entire embryo. So whatever my organs are formed, they are formed from inner cell mass, not the trophoblast. So they form entire embryo. Okay. Now there are cells of trophoblast. The cells of trophoblast, like these cells, which are present near the inner cell mass, they are known as cells of Robber. Every cell have a function, we will talk about them soon. So cells of Robber are what? The cells of trophoblast. Cells of trophoblast near ICM. What is ICM? Inner cell mass. Okay. So trophoblast is the layer that will help in the implantation. And what is implantation? Embedding of this blastocyst in the uterine wall. We will talk about that soon. But let's have a quick revision of all of this. First of all, after ovulation, oocyte will enter inside fallopian tube. It will meet the sperm and will do fertilization. By fertilization, zygote will be formed, which after 24 hours will start cleavage division, and the number of cell will increase. The embryo will be formed at 3rd day's morula, which have 8 to 16 cells, and it's compactly packed, but the size is same as zygote. Why? Because in cleavage, the cytoplasm never divides, okay, never increases. Now after mitosis, a 32 cell stage will form, which will escape zona pellucida and form a cavity inside it, and this stage is known as, very important stage, blastocyst. This is blastocyst, and blastocyst is 32 cell stage with a cavity known as blastocele, and this is the stage that will undergo implantation. Now let's see what is implantation. Now let's see what is implantation, a very, very important topic. So after the blastocyst is formed, this is like uterine wall. After the blastocyst is formed, this is uterine cavity. This is endometrium. This layer is endometrium. Now after the blastocyst is formed, the cells of Robber, they will come towards here and they will start dividing. Which cells? The cells of Robber. What are cells of Robber? These are the cells of trophoblast only that are present near the inner cell mass. So I am highlighting them with blue. This is inner cell mass. Now these cells, they will start dividing and they will form these finger-like projections. known as villi, So these structures are what? Villi, and you also call them as chorionic villi. Chorionic or chorion is an extra embryonic membrane. Like you must have heard of yolk sac. If not, I will let you know. There are certain membranes present outside the embryo which are not the part of main embryo, but they are present outside the embryo. They have a lot of functions to perform. Like I give you one example. One extra embryonic membrane is chorion. And chorions villi-like structure are known as chorionic villi. So it's the membrane present outside embryo. So that's why it is known as extra embryonic membrane. This has a role. What it will help in implantation? How? So it has formed villi. These villi, they will secrete certain enzyme, hydrolytic enzyme, which will erode the endometrium and make space in them. Just imagine this is endometrium. This is villi. Villi will keep on secreting the enzyme which will make spaces, in the endometrium, and they will start fit like this. And hence you say the blastocyst, with the help of its villi, it has embedded in the uterine wall, and hence this process is known as implantation. This is known as implantation. So after the villi has secreted a lot of enzyme, it will start to move. Inside the endometrium and make spaces like this, right? It will make spaces like this, and hence we say implantation has taken place. So one question that is most commonly asked, which embryonic stage does implantation? That is blastocyst. And what is implantation? It is the embedding of blastocyst in uterine wall. Okay. So this chorionic villi, it is extra embryonic, and what it is formed of? It is formed by cells of Robber. So who produces this cells of Robber? Or another question that can be asked, which cells out of inner cell mass and trophoblast forms the chorionic villi? It is formed by trophoblast. So formed by trophoblast, and the special cells are known as cells of Robber because these are cells of trophoblast only. Okay, and they secrete hydrolytic enzyme that erode endometrium to fix the embryo in it. And implantation usually occur at around 6th to 9th day. 6 or 9 not, it's up to like 6th to 9th day preceding fertilization, implantation takes place. Around 1 week after fertilization, implantation takes place, right? So that's how the implantation takes place. Now after implantation, endometrium is no more known as endometrium. You will start calling it as decidua. So this endometrium, I am writing it here, endometrium after implantation. You will not call it as placenta. Placenta is formed later on. Placenta is like 1 week after, okay, after implantation. So placenta is formed even after 1 or 2 weeks after implantation. Right now this is not endometrium, this because implantation has taken place. The endometrium will now known as decidua. So endometrium after implantation is known as decidua. Okay. Now, right now female is known as pregnant. Up to this stage the female is not pregnant at all. In most of females, up to this stage everything occurs, but implantation does not take place. That's why they don't get pregnant. This can be due to any reasons, okay. But up to this you cannot call the female as a pregnant because there is no sign of pregnancy in female. The sign of pregnancy in a female occurs when the implantation takes place. So when the blastocyst embed in the uterus, only then you call female as pregnant, right? So this is a very important point to note here, is that only after implantation, only after implantation, the female is said to be pregnant, said to be pregnant, right? Why? Because after implantation only, when the placenta is formed, the placenta secrete a hormone, and the name of the hormone is HCG, human chorionic gonadotropin. So after implantation, after the placenta is formed, it will start secreting this hormone HCG. Okay. Before implantation, the female does not have this hormone, right? So this hormone is detected in a test. You must have seen those pregnancy kits like Prega News, and so you have seen in the TV also, right? So these sticks, they confirm the presence of HCG in urine, right? If a female is pregnant, she will be secreting HCG. A non-pregnant female does not produce HCG because in her the implantation has not taken place. Imagine a situation if a female is not pregnant. If a female is not pregnant, there are two cases: either the fertilization has not taken place, or second, the implantation has not taken place. Maybe morula is formed, but implantation has not taken place. In that case also HCG will not produce. HCG will only produce after implantation, after placenta is formed, right? And there you call female as pregnant. There can be a chance that morula is formed and it will swipe away. There is no implantation. You will not call her as pregnant. So a female is only pregnant if she has HCG in her body, and that HCG from blood will go into the urine, and from urine that stick will detect the presence of HCG. So presence of HCG in urine confirms pregnancy. Okay, and the name of the test is Gravidex test. Okay, those sticks that come, that test has a name, that is Gravidex. Okay, so this is how a female is considered as pregnant. Before that, she is not considered as pregnant, right? Another point to note here is ectopic pregnancy. Now, what is ectopic pregnancy? We always say that the implantation takes place in uterus. This is what we say. In normal case, implantation takes place in uterine cavity. This is what we have done, right? But in abnormal case, or in case of ectopic pregnancy, the implantation takes place at any other place other than the uterus. It's not taking place in uterus but somewhere else. Let me give you an example: fallopian tube. Now for example, this is fallopian tube. Okay. In a normal female who is completely healthy and who does not smoke, and a female whose partner does not smoke, or the people she comes across does not smoke, in a way she is not taking smoke in her body. So the females who does not smoke, they have healthy cilia inside the fallopian tube. But a female who is a smoker, she does not have cilia in the fallopian tube. As a result, all those cells that we have made, the stages we have made, they will not be able to move. They were able to move. Why? They were entering into the uterine cavity because cilia was helping their movement. But what if the cilia is not present? All the divisions will take place in the fallopian tube, 32 cell stage will form in the fallopian tube, and implantation will take place in the fallopian tube. Imagine the implantation has taken place in the fallopian tube only. Now you will say, ma'am, what's the problem? Because uterus has that capability to take the gestation, because when the baby will increase, uterine cavity will increase. It has myometrium, it can expand, it can form healthy placenta. But when the embryo will keep on increasing in fallopian tube, the fallopian tube will burst and a female can die. So ectopic pregnancy is a pregnancy where implantation takes place other than uterus. Example, fallopian tube. Now what is the reason of this? Smoking. Smoking can destroy cilia. Smoking can destroy cilia, which will not allow the movement of embryo. Okay, and this can be fatal. This can be fatal. Fatal means a female can die. Why? Fallopian tube can burst. Fallopian tube can burst. Okay. So that's about ectopic pregnancy. So if the implantation is not taking place in uterus, you call it as ectopic pregnancy, and especially usually take place in fallopian tube. So the first scan of a female that occurs around 8 weeks of pregnancy, that is basically a confirmation scan to see whether the baby is in the fallopian tube or in the uterus, or whether the baby is present, if she is actually pregnant or not, right? So that's about ectopic pregnancy. Now let's see how twins are formed. You must have seen two babies are formed to a single mother, or a mother gave birth to twins, right? And sometime even 3, 4, 5, 6, 7, 8. I have seen like in a news 11 or 15 babies were born to a mother. So what happened at that case? So we'll stick more to twins and your concept will be clear for the others also. So what happened here? How the twins are formed? So twins are of two kinds first of all. One are monozygotic, also known as identical or maternal twins. Second are dizygotic, fraternal or non-identical. So you must have seen some twins which are exactly of like same face, everything is same. You just can't say, oh, are they different, same or whatsoever, right? So those are identical twins, they appear same, and you will see one pattern in identical twins. They are always of same gender. Identical twins will always be of same gender. Either they will be two female or born, or either they will be two male borns. They can be same or different gender. And even their characters, features will be different. Maybe the face is different, their face appearance is different, okay. So they appearances similar, their appearances may be different. Now why so? As their name suggests, okay, they may appear a little same, like a siblings, but not like that of identical, like that of the identical twins, right? Now what's the difference? You have seen here the word monozygotic. Mono means one, zygote. So what happened in the, how does identical twins are formed? When the zygote is formed, the zygote is dividing. Zygote becomes morula, morula becomes blastula or blastocyst. So when this blastocyst is coming out from zona pellucida, remember that stage when a 32 cell stage is formed and it is escaping from zona pellucida like this. It was escaping from zona pellucida like this. At this point, there can be a chance that the embryo gets split into two. There is splitting. When it is escaping from zona pellucida, let me show you in that diagram. Here, not here, here. When it is escaping from zona pellucida, there can be a chance this embryo gets broken down into two. So when it will get broken down into two, it has been formed from same zygote, it contains same chromosome, it contains same copy of chromosome, same DNA material. So these two embryo will be formed, will be of same gender, because this zygote, imagine the zygote was formed and it was like 44 plus XY, it was going to be a boy, and here all the embryos will also contain the information 44 plus XY. So that's why both these embryo 1 and embryo 2, it will also be 44 plus XY. On contrary, if we say, imagine the zygote was 44 plus XX, it was going to be a female. So embryo will also be both the females. So this is how, due to splitting of the embryo, the two embryos are formed. Hence it is known as identical twin, because they both contain same DNA material, right? So monozygotic, they are formed from same zygote, formed from same zygote, right? How it is formed? Formed by splitting of embryo, splitting of embryo, and they contain same DNA in both embryo. This is how the same twins are formed. What about these one? When a one male or another female is formed, or if both are of same gender, they don't appear same. So what happened here? Imagine in female, at same time two oocyte got ovulate. Like this is the how we form. There are two fallopian tube. The female have two ovary. You all know that, right? Usually what happen in a single cycle, one oocyte is released. What if due to some condition two oocyte are released? If two oocyte are released, two separate sperm will fertilize it. Two separate sperm will fertilize it, and two separate zygote will be formed. So this is your zygote 1. Imagine this contains information 44 plus XX, and this is zygote 2, which contains information 44 plus XY. So one male, one female will be formed. So here two babies can be seen. So here this is embryo 1, this is embryo 2, right? This is how fraternal or dizygotic twins are formed. So here two zygote form two different baby. So here two zygotes are released, and hence they are different. So they will contain different genetic material. I hope that makes sense to all of you now. Right? So that's how the twins are formed. Now let's solve some questions. But before that, let's read NCERT first. So the mitotic division starts as the zygote moves through the isthmus of the oviduct called cleavage towards the uterus. So when the zygote is moving towards isthmus, the cleavage starts, right? And form 2, 4, 8, 16 daughter cells called blastomere. The embryo with 8 to 16 blastomere is known as morula. So this is the most common question that can be asked, right? Morula continues to divide and transform into blastocyst. So sequence is also important, how it is formed. First you will get a zygote, then 2, 4, and then you will get 8 and then 16 cell stage, and then 8 to 16 cell stage is morula, and then what is formed? Blastocyst. So this sequence is also important, that can be asked in your exam, okay? Next, the blastomere in the blastocyst are arranged into an outer layer called trophoblast. The outer layer is trophoblast, and inner group of cells are inner cell mass. The trophoblast then gets attached to endometrium, and inner cell mass get differentiated into the embryo. So which layer forms embryo? Inner cell mass, right? And which helps in embedding into the uterus? That's trophoblast. As a result, after attachment, the uterine wall cell divide rapidly and covers the blastocyst. As a result, the blastocyst becomes embedded in the endometrium of the uterus. This is called implantation, and it leads to pregnancy, right? So you must be confused in this line. Ma'am, what happened after implantation? This will also be doing particularly in the next class, but let me tell you here also, once the implantation has taken place, the embryo goes inside the uterus, right? And when it will go inside the uterus, though I'll teach you this in the next class also, but because you will be reading NCERT, so I'll just let you know. After it has started entering inside the uterus will divide and cover the blastocyst like this, or I can say the outermost cell will cover the entire blastocyst like this. Or let me explain you in more detail. Like it has made villi, the villi is helping it in the embedding like this. This is stage 2. After that, the uterine cells will keep on dividing and cover it completely like this. This is what it is trying to say, okay. Alright, next. As a result, the blastocyst embeds in the endometrium of uterus. This is called implantation and lead to pregnancy. This diagram is also important because sometimes they will give you this diagram. You have to identify the cell structure. Like this is oocyte. This is fertilization happening here. After that, zygote will be formed. 2 cell, 4 cell. This is morula, 8 to 16 cell stage. 32 cell stage, and then blastocyst is formed. How you will get to know this is blastocyst? This has blastocele in this, okay. Alright, let's solve some questions. Read the following statement and mark the correct answer. The blastomere in the blastocyst are arranged into an outer layer called inner cell mass and an inner layer called trophoblast. The embryo with 8 to 16 blastomere is called morula. Choose the correct option from the following. So first is saying the outermost layer is of inner cell mass. No, outer is trophoblast, inner is inner cell mass. This is wrong. Embryo with 8 to 16 blastomere is definitely morula. So statement 1 is incorrect and 2 is correct. Answer is 4. Right? Next. The human embryo with 8 to 16 blastomere we have just done is morula. Answer is 1. Okay. Next. Which of the following differentiate into embryo? Morula, zygote, trophoblast, inner cell mass. So in morula the cells are not being confirmed which one will become the embryo. In blastocyst the cells have been differentiated into 2, the inner cell mass and outer trophoblast. And outer trophoblast forms villi, chorionic villi, which helps in implantation, and inner cell mass will form the entire embryo. So answer is 4. Next. Morula is developmental stage between zygote and blastocyst, between blastocyst and gastrula, after implantation, between implantation and parturition. So it's a definitely stage before implantation, so this is wrong. And it's a stage between zygote and blastocyst, right? Not between blastocyst and gastrula. You will study gastrula in the next class. This stage comes after blastocyst, and not between implantation and parturition. So answer is 1. So that's on a sequence, right? Next, match the following. Alright, let's do it. So column 1 says trophoblast, cleavage, inner cell mass implantation, and second says embedding of blastocyst in endometrium. So tell me, embedding of blastocyst is implantation. So for D we have 1. Group of cell that would differentiate as embryo, that is inner cell mass. So for C we have 2. Cleavage, mitotic division of zygote, that's 4. And trophoblast is outermost layer of blastocyst that attached to endometrium. So for that A we have 3, for B we have 4, C 2, D 1, answer is 2. Next, select the true statement with respect to morula of human. It has far less cytoplasm as well as less DNA than in an uncleaved zygote. It has more or less equal quantity of cytoplasm and DNA as in uncleaved zygote. It has more cytoplasm, more DNA than an uncleaved zygote. It has almost equal quantity of cytoplasm as an uncleaved zygote, but much more DNA. So it's a question on how nucleus and cytoplasm divide during cleavage. So as you know, the size of zygote and morula is same. Why? Because cytoplasm does not grow. So cytoplasm whatsoever is present in zygote is same in the morula. So it has almost equal quantity of cytoplasm but more DNA, because cells are dividing, definitely its nuclear material is dividing. Okay. Next. Cleavage is unique form of mitotic division in which nuclear division does not occur. No, nuclear division occurs, so that's wrong. Growth phase is absent, absolutely correct. No spindle developed to guide the cell. Without spindle formation, how will the division take place? Spindle formation is important, right? Plasma membrane of daughter cell do not separate. They separate. That's why they become 2 separate cells. You know, it's saying plasma membrane of daughter cell, it's same. No. They are saying the cells are fused. No, cells are not fused, they are separate. Answer is 2. So that's it about fertilization. We have already done in previous class. About implantation and cleavage, I hope you will study it really well and solve all the question, and we'll study the embryonic development and gestation in next class. Till then, just cover it up, learn it nicely, and I'll ask you questions in the next class also. And till then, take care, bye bye, lots of love.